And one of his main publications, many publications, he and his wife developed the blossom matrix. I just want to tell you that I learned this fact after I moved from Seattle, and it was one of the great discovery computational tools, I will say, that blossom substitute the block substitution matrix. Again, without a doubt, he's a true computational biologist, Dr. Henikoff. And please give a warm welcome to Dr. Henikoff as he discusses his latest work, the epigenomic landscape at the base pair resolution. Thank you so much, Dr. Henikoff.
Hey, well, thank you, Indika, for the kind introduction and for the invitation to come here and tell you about some of our recent work. I'll be talking about the epigenome. There's been a lot of excitement about it. I would define the epigenome as the DNA protein complexes that package our chromosomes, that package our genomes. Now, there's the mapping, the methods that people have been using for mapping the epigenome, however, have given us only a rather static view of the situation. And we've been interested in dynamics. And so I'm going to try to focus on the dynamics today. And I'm going to begin with the dynamic situation in which the transcriptional apparatus impacts the nucleosome. So what happens there? And then I'm going to move on to a more general way that we've come up with for mapping the epigenome dynamics. And I'm going to end with the least dynamic region of the chromosome, and that's a centromere, something that's been a long-term interest of ours. Okay, now we're going to start out with the nucleosome, and it's shown here as this sphere, but that's not anatomically correct. Rather, the nucleosome core looks like this. You have a tight left-handed spiral of proteins that the DNA simply follows around. And there's a central disk of histones 3 and 4 that's flanked above and below by dimers of H2A and H2B. Now, the bulk of nucleosomes are deposited behind the replication fork, so they go into newly synthesized DNA, but there's a set of histone variants that are replication independent in their assembly, so they go into an already chromatinized template. And if we consider those that are universal, or nearly so, there are 4. 2 of them are histone 3 variants, and 2 are H2A variants. Now, to replace H2A with, let's say, H2AZ, you have to partially unravel the nucleosome to make the exchange. But H3 is in the middle, and to replace it with H3.3, you have to unravel the nucleosome at least halfway, essentially evict the core in order to get the H3.3 in there. And therefore, we realized that we could gain a glimpse into nucleosome dynamics by profiling H3.3. We've done that in Drosophila, for example. This shows a gene-rich region, the map of a gene-rich region of the fly genome. And we see these H3.3 patterns. Now, it turns out where H3.3 is high, such as here, that turns out to correspond to sites, primarily of sites of active transcription, where the more transcription, the more H3.3 gets in. So what that implies is that when RNA polymerase goes through a nucleosome, it disrupts it in such a way that sometimes you lose a nucleosome that allow H3.3 to get in. Now that gives us a steady state view of the dynamics, and we wanted to actually measure the dynamics, the kinetics directly. And to do that, we came up with a metabolic labeling approach. We use an analog for methionine that goes into all proteins with methionine and has an azide group on it. Now we can derivatize that azide with an alkyne, in this case linked to biotin, such that we label all newly synthesized proteins with biotin. Now we're interested in the DNA that wraps around the nucleosome core. So we extract chromatin in the usual way. I'll talk more about that later. Then we wash stringently to remove the H2A, H2B dimers, and this will also pretty much strip off all of the bound proteins, such as transcription factors and the like. And then we can do streptavidin pull down of the newly synthesized H3, H4 core particles. We call this method ChEC. It was work of Roger Deal when he was a postdoc in the lab. And what you'll notice here is that the ChEC profile after a brief incorporation period looks a lot like the H3.3 profile, which confirms our assertion that H3.3 maps nucleosome dynamics because when we, nucleosome turnover, because when we look at turnover directly, we see similar patterns. Now, the relationship between transcription and turnover is correlative in this case, and we wanted to have some more direct evidence that actually transcription drives turnover. And to do that, Sheila Tevis in the lab looked at a region of the Drosophila genome where there are 3 HSP70 heat shock genes. And what you see here is what happens when we profile using chromatin immunoprecipitation, a method I'll talk a lot more about later, of RNA polymerase II. And what you see here are these peaks, 30 base pairs downstream of the transcriptional start site. This is the promoter proximally paused polymerase first shown by John Liss. And then when we heat shock, what we see is the gene bodies now fill up with Pol2 because there's immediate rapid transcription, a release of Pol2 going through genes. And if we look at turnover, what we see is that the gene bodies now show a dramatic increase in turnover. So indeed, this implies that active transcription drives nucleosome turnover. But we want to learn more about this. Like, what is it about transcription that is responsible for the turnover? And our attention fell on torsion. It goes like this. Think of DNA as being twisted in a right-handed manner, so it's positively twisted. RNA polymerase has to melt about 10 base pairs of DNA in order to start laying down the bases. Then it'll start moving forward. When it moves forward, think of it like a plumber's auger. It's forcing positive torsion downstream, negative torsion upstream. This is a so-called twin supercoiled domain model that's been out there for a long time. Now, the DNA that wraps around the nucleosome core I mentioned was left-handed, so the positive torsion would tend to unwrap the nucleosome. So we guessed that maybe torsion might be involved in this. So how do you measure torsion? So what we use is a compound that others have used, trimethylsorlin, which intercalates between the bases, and when you hit with UV light, it will cause a cross-link of the strands. And then Sheila came up with a method where we can precisely map the cross-link, and that'll tell us how much trimethylsorlin goes in. Now, when DNA is under-twisted, more trimethylsorlin will get in. When it's over-twisted, less will get in. So we can use this as a proxy for the torsion, and we can map it. And this is what we see if we line up all the genes at their 5' ends and at their 3' ends and show the average. The genome-wide average is the dotted line, and what we see is that upstream and downstream of the gene, we see an increase in negative torsion, and over gene bodies, we see positive torsion. And so that's the hypothesis of what we're looking at here. So to test that, we use an inhibitor of topoisomerase 1. Now, topo1 acts as a swivel to relieve the torsion, so when we inhibit it, we would expect to see increases in torsion in both directions, and that's what we see. So we see that there's an increase in negative torsion upstream and downstream, and an increase in positive torsion over the gene bodies. And that confirms the twin supercoiled domain model. Now, what about turnover? So we measured ChEC incorporation, use the ChEC method to measure turnover. And what you see here is that where positive torsion increases, we're seeing more turnover. Where negative torsion increases, we're seeing less turnover. And we see this not only for topoisomerase 1, which acts as a swivel, but also for topoisomerase 2, which carries out a breakage and pass-through reaction. So it relaxes the torsion, it relieves the torsion in a very different way, but we see basically the same kind of result. So what this implies is that when polymerase goes through, it forces positive torsion that destabilizes nucleosomes downstream and stabilizes them upstream, something Bill Gerard had actually shown in yeast on supercoils a long time ago, but we're seeing this in general over the Drosophila genome. Okay, so far we've been focused on the nucleosome, what's happening to the nucleosome from the nucleosome's perspective. I'm going to change the perspective now to that of RNA polymerase. So what happens when transcription goes through a nucleosome? What happens to the polymerase? Now, in order to answer this question, Chris Weber, when he was a graduate student in the lab, needed to come up with a way of, first of all, mapping RNA polymerase at very high resolution, ideally at base resolution, and also to be able to measure all RNA polymerases because what happens is when RNA polymerase hits an impediment, such as you might expect when it hits a nucleosome, by the way, RNA polymerase in vitro can't get through a nucleosome at all, it will backtrack, and when it backtracks, the 3' end will actually come out, and it'll come off track. And so the methods that are out there aren't really good for mapping that because they'll only map the engaged polymerase. So I needed something to get all RNA polymerases. The problem with doing things like ChIP is that RNA polymerase is very insoluble. But then Chris realized, well, maybe he could use the insolubility to purify the polymerase. And so that's what he did. And it's a very simple idea. And that is, we use a method that's been out there for over 20 years. We release a cytoplasm, nucleoplasm, and we wash the chromatin pellet, and all the engaged RNA polymerase will be there. So we'll have both the elongating and the ones that have hit the barrier and backtrack. So we'll get all of those in the pellet. And this just shows that we get quantitative recovery. This is of the elongating RNA polymerase. It's all in the pellet, essentially. And then we apply a published method for sequencing the 3' end of the nascent transcript. We call this 3' NT. And it's trans-specific. And here's what we see over a representative region of the Drosophila genome. What you're looking at here are these spikes. So here's an intron, here's an exon. And you'll notice that the density of the spikes is very similar in introns and exons. That's what you'd expect if what we're measuring is the base that's being added onto the growing chain of RNA in the active site of RNA polymerase. So this is the ultimate map of transcription. All the processing events happen after this. Now, what we want to know is where are these spikes relative to the nucleosome landscape. So we can map the nucleosome landscape using MNA-seq. I'll say more about that. We digest the unprotected DNA and we do paired end sequencing, and it gives us a nucleosome landscape that looks like this, shown here on both strands. So then the question is, where are these spikes relative to the nucleosomal peaks? It's just a blow up. And so the question is, where is this spike, for example, relative to the dyad axis of the +1 nucleosome? So what we're going to do is we're going to use a coordinate system based on the +1 nucleosome, line up all the Drosophila genes at their +1 nucleosome, and then we'll rank order them by the amount of 3' NT signal, the amount of Pol2 that we see, and it looks like this. So here's a heat map representation, and what you'll first notice is that the vast majority of the stalls occur 73 base pairs upstream, the entry site of the nucleosome. You'll also see that the ones with the most Pol2 turn out to be the promoter proximally paused polymerases that I pointed out earlier, because they're at +30. But it turns out that when we separate those out and look, they also will stall when they hit the entry site. So this is a housekeeping process. Essentially, all RNA polymerases will stall to some extent when they hit the entry site of the nucleosome. Now, this is just another representation of what I just showed you. So here is the profile of all of the +1 nucleosomes, and here is the stall density, and you see it's at the entry site here. When we go to +2, we also see a peak at the same place, and when we go to gene body nucleosomes farther in, we also see stall here, but we see some encroachment into the nucleosome. If we ask where this maps on the nucleosome itself, what we see is that there's some encroachment. So RNA polymerase, the active site, lies about 15 to 20 base pairs from the front of the polymerase as it's going through. And so what we see is that it's actually starting to unwrap the nucleosome when it gets in. And that's happening at essentially all nucleosomes. What's striking about this is that that's not where it's been mapped in vitro. There are many studies in showing that in vitro, the stall is somewhere else. Well, I told you in vitro it can't get through. You have to add something else to get in there. But in vivo, it's a very different place. Our interpretation is that this is where the histone DNA contacts are the strongest, and that's why you see it in vitro. But in vivo, there's a lot going on there that's allowing RNA polymerase to get through this barrier. And what might that be? We first look at the magnitude of the stall, and we see the +1 nucleosome shows the biggest stall. The other stalls are remarkably uniform, but much lower. Now, we realize that the +1 nucleosome is special in a number of ways, and from our perspective, the most interesting difference is the +1 nucleosome is really loaded with that histone variant that I mentioned at the very beginning called H2AZ. So H2AZ is universal, is universally found. It's been a puzzle as to what it actually does. But finding this kind of situation, we wondered whether H2AZ is involved in this housekeeping process of stalling. So first thing we did was to map H2AZ occupancy, and what we noticed was if we plot the occupancy as a function of the stalling, they're anti-correlated, as if H2AZ is acting to reduce the stall. To test that, we knocked down H2AZ, and we measured the 3' NP signal and the remaining H2AZ, and we divided up nucleosomes into whether there's no change in stalling upon knockdown or increased stalling upon knockdown. And what you see for +1, +2 in gene body nucleosomes is that where there's increased stalling upon knockdown, we see less H2AZ, which confirms that H2AZ is really reducing the nucleosome barrier, because where there's less of it, you get more stalling. So what this, first of all, implies is that actually H2AZ has evolved to reduce the stall. And that could be, in general, so the stall is a big problem for RNA polymerase to get through, and H2AZ has evolved to reduce that. What it doesn't explain is, why is there a stall in the first place? I mean, why isn't that the +1, why should it be different from the others? And I can only speculate on that, but here's the way we're thinking of it. If you go back to the data I showed you previously about torsion, we see that the +1 nucleosome is different from the others downstream in that it doesn't respond to torsion. In other words, we don't see any increased turnover when we increase the torsion by inhibiting the topo. And so what we infer from this is that the +1 nucleosome is inherently resistant to getting removed by torsion. Now think of the torsion as being the tool that Pol-2 uses to get the nucleosome out of the way, but it can't build up enough of a head of steam in order to actually get that +1 nucleosome. It's sort of the physics of the situation as such. That's the best it can do. So it needs something like H2AZ to make the nucleosome come off more easily would be one thing. But we also think that the fact that nucleosome remodelers, histone modifications of a particular types, tend to be loaded over that +1 nucleosome. We see that as an adaptation for allowing Pol-2 to get through that +1 nucleosome. Now, what I'd like to turn to, so that's one kind of dynamics, the dynamics between RNA polymerase and the nucleosome. Now what I'm going to turn to I'm going to turn to methods for looking at other components of the epigenome where we can we'll eventually get to the dynamics. But first, I want to introduce you to actually an enzyme that's going to be our favorite tool for doing this, and it's called micrococcyl nuclease. Now, those of you who know about chromatin work will be familiar with micrococcyl nuclease because it's been used for decades for mapping nucleosomes. Now, eminase is a single-strand specific secreted glycoprotein. Now, you might wonder, well, how does it cleave double strands? Well, we're not actually sure, but what we think happens is DNA breathes a little bit, allows eminase to cleave one strand, making it easier to cleave the other strand, and then it'll nibble on the ends until it reaches an impediment, such as a nucleosome core particle, and that's why people have used it for mapping nucleosomes. But we realized that eminase would probably stop nibbling anytime it hits a DNA binding protein, so that we modified the Illumina protocol for paired end sequencing such that we could recover fragments that were 25 base pairs or more, and then we would just map and stack the reads. Real simple. And this is a representative region of the yeast genome and the intergenic region. And this shows an M&A series where all we're mapping are the smaller fragments. The larger fragments tell us where the nucleosomes are. So you see these phased nucleosomes down here. Now the smaller fragments, we see this whittling down of what looked to us like maybe 3 particles. They're close to one another. Each protects about 30 base pairs, and they show different occupancy. Now this is a general method. We applied it, for example, to Drosophila. And when we look at the small fragments and do the mapping and stacking the reads, and look at that HSP70 region I showed you previously, we see that there are these squarish peaks there. This one corresponds to GAGA factor. This one corresponds to TATA binding protein. From previous work, we knew that they were there. So we have a very nice method for profiling the epigenome in general. Now, we realize that we get the same profile if we extract the DNA immediately after the MNase digestion, or extract the soluble chromatin, then extract the DNA from that. And we realized that we could use this soluble chromatin as the input for chromatin immunoprecipitation just by pulling it down with an antibody for a particular, let's say, transcription factor. So I'm going to illustrate this strategy using a transcription factor from yeast called Reb1. Now, Reb1 had been mapped by Rick Young's group over a dozen years ago using cross-linking and sonication, so-called ChEC, and the readout was on microarrays. And then that was followed by calling motifs using standard method, and that could identify sites, specific sites in the genome. And this is just a representative region showing that there are 2 sites using this method of Reb1 occupancy. Now, a big improvement to this protocol came from Frank Pugh, who basically resected back with an exonuclease, get high resolution and low background, and this allowed him to do find many more peaks, and it was quantitative. And in addition, he didn't need to use the motif calls because the peaks were sharp enough. Okay, so we wondered how this simple method that I just described, where we pull down from this solubilized chromatin, how this would fare against it. So what we do is we call this organic for occupied regions and genomes from affinity purified naturally isolated chromatin. The idea of the acronym is that we're not adding any toxic compounds such as formaldehyde. And when we map the small fragments here, this is the raw data you're looking at here. We get pretty much the same peaks you get with ChIP-exo, and then some. We get a lot more peaks. And if we use the mean program to basically identify the motifs that are represented by these peak calls, we see we get the same Reb1 motif in both cases. Now, it's worth noting that for organic, it's very, very specific in the sense that 99.3% of our peak calls have the motif. So it's very highly specific, much more specific than the other methods that we've used. Into a limitation that we ran into of this organic method is that when we got into higher eukaryotes, when we got into large complexes such as RNA-Pol2, we found that solubility was a limitation. We just couldn't solubilize everything, only a small part, and that worried us. So Pete Skeen, a postdoc in the lab, came up with the following method, a high-resolution cross-linking chip method. So we do formaldehyde cross-linking in the usual way, and then instead of sonication, we just do MNase digestion. Now this is insoluble, but with brief sonication, we can get it out of solution. So CHD1 is a nucleosome remodeler that Pete was interested in studying, and what he found was that with just brief sonication, you go from poor recovery to quantitative recovery of CHD1. When we applied this to RNA-Pol2, which I told you was terribly insoluble, what we basically see is that we get a result that really nicely corresponds to 3' NT, where we've mapped precisely where RNA-Pol2 is. So this seems to work. If you're wondering about what's wrong with conventional ChIP, you're basically seeing it over the transcriptional start site. We think that's a sonication artifact. So just be aware that as usually practiced, that chip has some issues. But it looks like this high-resolution XChIP works pretty well. So we applied it to transcription factor, in this case CTCF, so we can compare it with other data. This is from human cells, and you see compared to the ENCODE profile, which is kind of broad, we get a very nice sharp peak here that's right over the CTCF motif. It also compares very favorably with ChEC. So this is another nice method, but in both cases of these new methods, it's giving us a static picture, and we really want to know about the dynamics of transcription factors. And for that, we've abandoned ChIP, and we've come up with a different approach. Now, I'd like to say this is new, but it's not. This is something that was published by Uli Lemley over 10 years ago, and he calls it Chromatin endogenous cleavage, and it goes like this. And we just added a sequencing readout. Everything else came from Lemley. So we make a fusion between our chromatin protein of interest, our transcription factor, and micrococcal nuclease. Now, micrococcal nuclease is entirely dependent on calcium, and intracellularly there's very low calcium. That's true for yeast, that's true for us. And so all we need to do is permeabilize the cells, add calcium, and just wait while the MNA starts to cleave. It's already bound if it's a transcription factor. Then we simply purify the DNA. We use an ampere bead selection step, very simple, to enrich for small fragments, and we make libraries. In other words, we take cells that are alive, we add calcium, and a few moments later, we extract the DNA and we make libraries. That's it. The advantages include it's in vivo, cells are alive, there's no chromatin solubilization, no antibodies, no fractionation. This is very much like a method that Bas van Steensel, when he was a postdoc in my lab, came up with called DAMID, where he tethers DAM methylase. But in this case, we're using MNase itself. And the advantage over DAMID is that, first of all, it's controlled exquisitely by calcium. And as I'll show you, we can get near base pair resolution. Here it is for Reb1. And this is work of Gabe Zentner, a postdoc in the lab. Now, this is the Reb1 motif on the opposite strand from what I showed you before, and we're doing paired end sequencing, so we get the cleavages on the left end and the right end independently. With no calcium, we see this background here. Now, 10 seconds later, after calcium addition, we see dramatic cleavage. This is for all 2,000 Reb1 sites. We see dramatic cleavage on both strands. So you get double-stranded cleavage over here. Now, 20 seconds. Oh, so we get orientation, you might notice. Turns out that this is what we see if we put MNase on the C terminus. If we put an N terminus, it actually cleaves on both sides. Now, 20 seconds. Now we see a difference. So what we're seeing is the beginning of the nibbling. And now you see these sharp peaks, 30, 40, 50, 1 minute. And you can see that the peaks move closer and closer till we're within maybe a base pair or two of the Reb1 motif. Now, if we go longer, what happens is that everything begins to drop down as if what's happening is the transcription factor is actually releasing the DNA, and then it keeps going down out to 20 minutes. Here's an animation of this, what I just showed you, so you can see the double-strand cleavage, the nibbling, it goes up and it goes away. Now, how do we analyze data like this? So what we did was to pool all the time points and do the peak calls on the pooled time points. What we found is we got a lot more peaks than if we just looked at just a short time point. And we realized that these might fall into two categories. So we just did clustering. K means clustering, K equals 2, basically. This is what you see. So we have one category, which is the fast sites, and they peak at 40 minutes, and then they go down to nothing. And then we have another group that takes a long time. They're very slow. Several minutes pass before we begin to see something. And then if we ask who has the motifs, it's almost entirely the fast sites have the motif. The slow sites don't have the motif. Now, this is general. We did this with ABF1 and BRAP1, two other yeast transcription factors, and we see the same thing. ABF1 is particularly good. It peaks at 30 seconds and then goes quickly down. Now, our first thought was, well, the slow site, well, we know what the fast sites are. That's the same thing we pick up with ChIP. The slow sites, though, presented a problem. What are they? And our first thought was, well, maybe it's just random background. If there was random background, then we should, then first of all, so I can tell you that this is very repeatable. We get the same sites for the same transcription factor. But when we look between transcription factors, we find that the large majority of them are unique. So that implies that the sites that we're seeing are specific for the transcription factor, as if there's some recognition there. So let me just give you a model for what we think is going on. We think that the fast sites would be the standard sites where the transcription factor is bound, and it spends a long enough time there that it can cleave. Now the slow sites, we think, are the transcription factor looking for its site. So it's probably going by one-dimensional diffusion any place within the genome and just kind of scanning along. So it never spends enough time at any particular site to give it a high probability of cleavage. But sometimes it will find something it likes, doesn't have the motif, but it likes it, and it will cleave it, and it will cleave it reproducibly. And we have some evidence that this might be the case, and I'll just outline what it is. So if we compare non-crosslink to crosslink, like organic to the cross-linking chip, so we did that for ABF1, what we basically find is that there's not much effect on the fast sites, but of cross-linking, as you might expect if it's always there. However, the cross-linking increases the frequency of slow sites and also increases the frequency of random sites. So it suggests to us that we're getting time-resolved mapping at base pair resolution of the transcription factor bound to its site, but we're also detecting this scanning process that's occurring. So we're capturing the dynamics in a way that you can't get with chromatin immunoprecipitation. We're still working on getting so we've only done this in yeast, but we're trying it out in other organisms. As far as we know, it should work just fine, but we haven't done that yet. Okay, so I've been talking about dynamic regions, about dynamic parts of the chromosome, basically where the genes are. Now I'm going to turn to this rather static region of the genome, the centromere, which is embedded in heterochromatin, and nothing much goes on there during the cell cycle until mitosis, and that's where the centromeres are used. That is, the centromere is the site of formation of the kinetochore, the proteinaceous complex that attaches to the microtubules to pull the chromosomes to the pole. So it only has one job, and that job is out of sync with everything else that's going on when the rest of the chromosome is completely condensed. Now, the reason that we like to focus on the centromere is it's long been known that there's a centromeric histone variant, we call it CnH3 generically, but it's not quite universal, but it's almost universal. It's ancestral for sure, and it's a special centromeric nucleosome, very different, histone, very different from histone H3, from histone H3.3. Now, and I'll be calling it, we're going to talk about it in yeast and in humans. In yeast, we'll call it CSE4, is the traditional name, and in human, centromere protein A, the traditional name there. Now, we'll start out with yeast, and the reason we like to study it in yeast is that it's long been known for some 30 years that yeast centromeres are determined by DNA sequence, and it was known for 30 years that it's a tripartite sequence, variously referred to as CDE1, 2, and 3. Now, CDE1 is the binding site for the CBF1 protein. CBF1 is like REB1, ABF1, RAP1. It's a general DNA binding protein, so it binds a lot of other sites, and it's non-essential. However, CDE3 is the binding site for a kinetochore-specific complex that is absolutely essential and is needed for localizing the CSE4 nucleosome. So we can ask, where is the CSE4 nucleosome? You might expect it would be right here over CDE2, and you'd be right. So we do organic profiling for all 16 yeast centromeres, and this just shows we get a very high dynamic range when we look at the ratio. And when we go through our M&A series, you see that there's a shelf over CD1, a shelf over CD3, and you can see that the centromeric nucleosome lies right in the middle. In fact, depending on how we map this, we can show that there are actually 3 distinct particles here. So it's only 80 or so base pairs of DNA that wraps around this particle, indicating a single DNA wrap. Now, we wanted to know where this particle, how this particle is oriented within CD2. To do that, we adopted a method that was introduced by the late John Whittam. John was actually collaborating on this project, and when it got started, and what John came up with was a method for turning histone H4 into a cleavage reagent, a site-specific cleavage reagent. And it goes like this. In red is serine 47. You change that to a cysteine. You derivatize it with a phenantholine compound that will chelate a copper, and the presence of hydrogen peroxide will initiate a strand cleavage reaction. And this was modeled by Srinivas Ramachandran, who's a postdoc in the lab, and he came up with this structural model. What goes like this. So the copper's in blue, and every time it comes within 4 angstroms of the bond that it can cleave, you see a dotted line. So you can see that there are cleavages on the Watson strand and cleavages on the Crick strand. And they map like this. So this H4, we'll put these cleavages in there, and this H4 we'll put in these cleavages. This is predicted, and we can compare that with what we see genome-wide in yeast by taking all 60,000 yeast nucleosomes and basically deducing where the diet axis is and lining them up. And what we see is the cleavages correspond precisely to what the model says. So we think we know how this reagent actually works. Now, this is what you see over the entire chromosome 3. And we're going to focus our attention on the centromeric region. And what we see is that as we get closer and closer to the centromere, you can see that there's somewhat, some sharpness, but mostly fuzziness around the centromere. We actually see that genome-wide, but we see a very distinctive pattern over the CDE2, over the centromere, distinctive pattern of cleavages. And in fact, we can actually show that the patterns over all the centromeres are different from what you see at any other nucleosome. So they're definitely distinctive. When we line up all 16 yeast centromeres, what we see is that there are these 4 peaks. There's a cluster of peaks that are 10 base pairs apart over here, and over here they average about 43 base pairs apart. And we can ask how, and this is where it lies within the ChIP signal that I showed you. So it's right in there over CD2. Now we can ask, how does this fit with models that have been proposed for the centromeric nucleosome? There's the octameric model, the conventional model, also a model in which you don't have H2A or H2B, but you have 2 each of CSE4 and H4. And these models don't fit. They would predict cleavages in the middle, but more importantly, there's too much of a wrap over here that would be protected to account for this peak here, which is too close to the edge of CD2, CD3 junction. So these models are incompatible with the data. However, what is perfectly compatible with data is a model that we'd actually proposed earlier called the half nucleosome or hemisome model. And that is that you have within a population of cells, you have equal numbers of these four structures, where you have 2 different rotational positions in this orientation and 2 different rotational positions in the opposite orientation. I think that's pretty remarkable if you think about it. Here we have this asymmetric sequence, asymmetric proteins. However, the orientation of the protein is very flexible within there of the complex. So that gives us a very fine structure map of yeast centromeres, and so what we can see is that they're constrained by a single wrap, and they have DNA binding proteins on both sides, sort of holding them in and allowing for these hemisomes to form uniquely at this site on the chromosome. So then the question is, well, how well does this hold for other centromeres? So we're going to go to a complex situation, and that's the human genome. So those of you who are aware of the human genome, you probably who know about this, are probably aware that the human genome is not even close to being completely sequenced because all of our centromeres are gaps. There's no connectivity. You have multi-megabase arrays of this homogeneous 170 base pair alpha satellite repeat. But there's some structure on the edges in the paracentric region that consists of these repeats, but show this very interesting structure. So you have monomeric 170 base pair repeats that are quite diverged from the majority of the alpha satellite. And then you have these higher order repeats that show this old to young structure. So let me explain what I mean by that. If we look at the X chromosome, we find either edge of the X chromosome, we find these 170 base pair repeats present in 12-mer units. And the sequences within the unit are similar to one another, but quite diverged from one another. And when we go from distal to proximal, we find that they get more and more homogeneous as we get closer. We infer from that these are older and these are younger because basically these guys have diverged from these. Now the question is, what's in between? Well, because you can only sequence short fragments, in this case Sanger sequencing of 800 base pairs or so, if they're homogeneous, you don't know how to align them if they're perfectly homogeneous. So there's these nearly identical unassembled repeats in between. Now, some have argued that, oh, well, it must be the same thing on the edges as in the middle. That's what you might think. For reasons that I can go into in the discussion, we don't think that's true. And as a result, we're very skeptical. And so we thought, well, maybe there's another way that we can get at the human centromeres, and that is to use chromatin IP, to use ChEC, to identify the centromeric sequence by function. So we basically pull it down, and now we've got a bag, now we've got all these sequences of functional centromeric sequences, and we can see if we can make any sense out of it. Okay, that's the idea. How do we do that? We first of all do ChIP. This is work of Jitendra Thakur in the lab, postdoc. And we first pull down CEMP-A and centromere protein C, another intercentromere protein, and we get the same result for both. And this is pretty much as we expected. So what we see is that there's high density of the centromere proteins where the sequence is fairly homogeneous, fairly young, and then it kind of poops out as you go in, as it diverges, because it gets farther and farther away from the sequence. And we see that H3 is reduced, as you would expect, because CEMP-A is basically taking its place. Now, that's what we expected. What would be much more interesting would be, well, what about what doesn't map here? What do we get that doesn't map there? Here's how we deal with it. So we have 100 base pair reads. We can merge them, and as the following way. So we only want those that cover a single unit, so we can be confident of what the sequence is. And so we get everything from 25 base pairs up to 125 base pairs. Now, when we pull it down, we basically get almost entirely alpha satellite. That's good, but they're diverged anywhere from 100% identical down to 60% identical. We don't know. So we basically apply a greedy algorithm that can deal with millions of these reads, and we just cluster them. And we just cluster based on sequence similarity, being very stringent. We don't want to put anything together that's not really part of a cluster. And we get about 200,000 clusters from the human genome, so that's a lot. However, it turns out the vast majority of them are singletons. We don't really care about them. We only care about the most abundant ones, because those are the ones that we figure are the ones that should dominate human centromeres. So we rank order them based on the size of the cluster, and then we're just going to arbitrarily look at the top 20. And to represent these sequences, I just show a phylogeny. And what you see is that there are these 4 branches. Now, when we use each of these 20 sequences in Search GenBank, what we found was that these two branches correspond to what's been annotated as being part of chromosome 1 and some other chromosomes. And these two correspond to chromosome 13 and some other chromosomes. So we'll call this chromosome 1-like and this chromosome 13-like. Now, there were also several unplaced clones, and these are the really interesting ones. These are the ones where, and by the way, this is all from the Craig Venter genome, although we've also done this with 4 other genomes that basically see the same, get the same phylogeny. So this is going to be true for the human genome in general. Now, we wondered about these unplaced clones. So the basic thought is they're unplaced because you could partially assemble them because there's enough divergence there that you could assemble them, but they're different enough from what was on the edge of current assemblies that they could be put in there. And there were several of them, and I'm going to show you the best, and that was this one here. So if we take number 1, sequence 1, the most abundant, we see that it maps at 11 places with 90% average identity. Number 2 maps at 10 places, 96%, etc. Okay? So that's for this branch. And it turns out this branch matches right in between. So we have, you can see this dimeric unit, average about 95% identity. Now, we had a way of checking this dimeric unit, and that is that it's well known that there's a protein called centromere protein B, CCMB, that is a DNA binding protein with exquisite specificity for a 17 base pair, 15 to 17 base pair binding site. And it only binds to centromeres, as far as we know. And it's the only DNA binding protein in centromeres. And we find it's called the CCMB box, its motif. So we find that motif every 340 base pairs. So we have this very precise dimeric unit of the CEN1-like consensus. Now these are the most abundant sequences, but it turns out it's also abundant in another way. So we compared the ChIP abundance within our libraries for everything that has been annotated as alpha satellite. We find that CEN1-like really is the most abundant there, accounting for 38.4% of the total. We also analyzed PacBio reads and were able to deduce that that's a pretty good estimate of the total in the alpha satellite. So this is the most abundant sequence, and it turns out that CEN13-like is also almost similar. It's very similarly abundant. Looking back in the literature, we found that actually this was noticed before, that there's this dimeric unit that's present on these 9 chromosomes and one present on these 11 chromosomes. So we've accounted for all but 4 chromosomes. So we basically have the sequence that dominates the human centromere. So the question is, what happens if we map our reads to this? So this is the CEN1-like, and what we see is a very striking structure. So we see a pillar of 100 base pairs here, a pillar of 100 base pairs here, with a gap in between. And we pull down with CEMC, we see the same thing. But now you can see that the CEMP box really shows up. It's known that CEMC interacts not just with CEMP, but also with CEMP. So what's happening is it's pulling down the whole complex that's got A, B, and C all together. Now, if we look at the paracentric regions, these higher order repeats that most of the attention has been focused on before, we see they don't show the structure at all. So they're really diverged, and they don't show, they basically, they have a totally different structure. So this is for the young human alpha satellite sequences that dominate our centromere. In fact, the positioning is so precise, it's even better than what I showed you for yeast. So if we look at the homogeneity of these sequences, they're all just right around 100, almost all about 100 base pairs. So we see this 99, 90, 97, 99, 101, 103. So that indicates that what we have is a particle that gets nibbled a little bit by emanase, but it really likes to hold together. There's not much encroachment of the emanase on this. It's a very tightly packed particle. And that's for CEN1-like. CEN13-like, which is the other dominant one, is 2 base pairs bigger in a repeat unit, and it's also 1 base pair larger with respect to the minimal size. Our interpretation is that this pillar here and this pillar here are both 1 base pair longer for CEN13-like. So finding these very specific sequences dominating most of our centromeres, we basically conclude that human centromeres are not epigenetically defined, as some have argued, but it's defined by sequence, very much like what I showed you for budding yeast. So I think that most centromeres are going to turn out to be like this, that they will basically have these single wrap particles that will dominate, that would be just single sequences. So it's comforting that actually sequence does matter. Actually, I would say it's as genetically defined in humans as it is in budding yeast. So what's in between? We looked at another centromere protein called centromere protein ChEC, which we were not able to pull down using native, but we used this cross-linking chip method that I mentioned earlier, and now we see that ChEC pulls down the same particle, and we would deduce that it fits right in between. Now, what is ChEC? ChEC is a histone fold protein. Together with W, S, and X, forms a tetramere, a tetrameric particle that will wrap DNA. And Tatsufuku Gao showed this, also showed that it induces positive DNA supercoils. And that was reminiscent of something we'd shown many years ago for budding yeast in vivo. We showed that centromeric nucleosomes induce positive DNA supercoils. So let's go back to the beginning of the talk, where I talked about torsion and the orientation, the wrap of the DNA left-handed around the particle. Well, what we infer from this and from our mapping is that both of these particles actually wrap DNA in the opposite direction. So it's a fundamental distinction between centromeres and the rest of the genome. It wraps DNA in the other direction. Here's our model for it. So we think that the DNA will wrap 100 base pairs around the particle in a right-handed manner, go around the TWSX part, and continue on, and continue on here, and this would cover the 340 base pairs. We think that they should stack, because we know nucleosomes from structural work like to stack, so that's our model for it. One nice thing about this model is that the connection of the chromatin to the outer kinetochore is in two places. One is ChEC, and the other is ChEC, and both of them are facing in the same direction here. Okay, but why the right-handed wrap? So I'm going to speculate on this, and it comes from the fact that when you pull on DNA, forget nucleosomes, just pull on DNA, allow the ends to freely rotate, it'll actually overwind up to about 30 piconewtons of force, and then it'll unwind, come apart. Now, that overwinding is interesting from the point of view that if you force positive, you're forcing positive torsion on what you're pulling, so that would unwrap nucleosomes, and that's not good for these guys. You really want them to wrap. So the basic idea is that they would tighten up. So here's my molecular model for it. So at Interphase, we have these particles here, these pillars, and you have this TWSX over here. And one of these guys is going to, ChEC and ChEC, are going to interact with the outer kinetochore. So there's a high probability of one of these guys getting caught and getting pulled. You pull on it, and everything will come together, so you have a nice platform to pull on. So that's what we think that the right-handed wrap is an adaptation towards making a platform to pull on rather than anything that you can transcribe through. Okay, so I'm out of time actually. So I'll just summarize. I described, first of all, ChEC, which is a method introduced by Roger Deal with his own lab at Emory University, showing that transcription drives turnover, from Sheila Tevis, who did the torsional work and introduced TMP-seq, 3'NT for mapping PAL2 and showing the stalling is work with Chris Weber. Organic profiling is work of Siva Kasanathan and Gabe Zentner, and Gabe also did the ChEC work I showed. H4 cleavage work is involves a number of people, including Christina Krasowski, former graduate student, and Srinivas Ramachandran. And the clustering work, the human centromere work is basically, the computational work was done by George Anikoff, and Jitendra Thakur did the wet lab work, and she also did that work on the CEMP complexes. And Pete Skeen is the one who introduced the high-resolution XChip. And these are our funding sources, and I'll stop there and take any questions. Thank you. Tom.
Well, Steve, there's a lot to discuss, like always, after your talk, but let me focus on the dynamic issue. And I think that focusing on dynamics is important because it is ignored a lot. In your assays, I mean, one of the reasons we use crosslinkers is to try to counteract dynamics after cell lysis or disturbances. And I'm trying to think in your assays how you can exclude that some of the patterns are produced after the cell has been disturbed. Have you tried to look at any known dynamic processes where the binding pattern changes in response to a signal to measure that rate of the change in your, quote, in vitro, in your permeabilized cell system to try to evaluate whether the dynamics in that assay represent what we think we know about dynamic changes in transcription or other processes?
Well, the ChEC that I showed you is it's an independent way from ChIP. It's totally independent from ChIP, but we were pulling out the motifs. So we look at just the short time points. They correspond to what we see by ChIP quite remarkably well. So at least in yeast now, I can say with confidence that we weren't being fooled by the. Oh, and the other thing is that I showed you both for crosslink. So for ABF1, I didn't show the data actually. We get the same result whether we crosslink or not. So that's sort of 3 ways of doing it, and we get the same result. The first approximation, but it's a good point that we often don't have any. We can't be sure of what's going on with whether when we don't crosslink, except that one thing to keep in mind is that when micrococcal nuclease very rapidly will get rid of any DNA there. So if you imagine a protein moving or coming off, it's going to chew up the DNA. So it's very unlikely, I think, for it to come off and then rebind or move around. So the micrococcal nuclease should take care of that. I can't prove it, but it seems very likely.
Any other question?
This is just a short question, but do you think that the centromeric sequences are conserved between species?
That's a really good question, and we've been looking at that. So I can tell you so far. So we've been analyzing data. Now we've been pulling all the primate data out of the databases and looking at. So we've been looking for these 2 sequences, the two dimeric sequences. We see the dimeric sequences going all the way back to macaque. We see the same 2. Beyond that, we don't. So we think it's somewhere around there. I don't know in time, but they are conserved. Yeah.
What I understand is that, in the mouse cells, like alpha satellite sequences are more like uniform compared to the human cells. So
you mean the centromere?
Yes.
Yeah.
And also pericentromeric heterochromatids.
We're looking at mouse now. That's really interesting. So mouse minor satellite is 120 base pair repeat that is thought to be very uniform.
Right.
But remember, there's this ascertainment bias. You can't assemble it, so you're making an assumption that it's all going to be the same. And I don't think that anybody's really looked at critically, so we're doing that now. But I don't have an answer. We haven't gotten that far yet.
One question about the transcription factors. You showed that you have very strong binding sites with the MNAs, but then you have others that are longer time binding.
Mm-hmm.
Are those potentially sites that do bind transcription factors but haven't been primed for transcription?
I'll tell you what we think, and we're currently testing this. What we think that there's sites that the transcription factors like, but it does, but they don't have the motif. And here's how it might work. So Arima Rose at USC has found that he can improve motif prediction. He can DNA binding prediction. He's shown he can do that by taking into account shape parameters. So essentially, it goes like this: when you have a motif, you're only looking at one base at a time. But shape is determined by nearest neighbor kind of interaction. So the shape would include the width of the minor groove. AT tends to be very narrow. Okay, things like that. Also the tilt of the bases and the twist. So roll, it's called. So using those parameters, he can find consistencies for binding that don't show up in the DNA sequence. So I just got an email from, we sent him our sites, and he said, "Oh, looking promising." So that's all I can say. So our working hypothesis is that it's shape that they're really recognizing when they have these preferences. It's not enough to really hold it down because you really need the right sequence for that.
How do you think neocentromeres evolve regarding those sequences?
Yeah, that's a good question, because I was making a big point of it being genetic. I would just point out that all I'm talking about are our native centromeres, that the vast majority of centromeres, but there are a few. There's 1 in 10 million people, or 1 in a million, have neocentromeres that don't correspond to alpha satellite at all. And as far as we know, they're just perfectly okay, except they have never made it through evolution. So as far as we know, there's I mean, going back to over monkeys, there's not a single case where there's a neocentromere that anybody has seen actually evolve into a regular centromere. So I would first say that neocentromeres, they do exist. They're very, very rare, and they're not terribly successful. So I think what we're looking at here is a highly evolved situation, and you can understand the reason it would be highly evolved. You really want to make your centromeres perfect, because one error in 10,000 is not going to make it through evolution. You've got that cell division has to be perfect. You can't be missing chromosomes. So I just think that what we see is an optimized situation where you have these perfect positioning to get perfect for it to be perfect. But neocentromeres are examples where that doesn't happen, and they're epigenetic. I fully agree, but they're so rare that I don't think we really should focus so much attention on them. So what you find, what I'm counteracting, is the fact that just about every paper you read about human centromeres will basically say, or centromeres will say, oh, centromeres are epigenetically inherited. But that's all based on these rare events or on artificial chromosomes, which don't really work very well. But I would say that once you actually see what's there, it's remarkably precise. Yeah.
So the structure of centromeric nucleosomes has been discussed a lot, and I don't know that we need to rehash that here, but I'm curious whether your model, the dynamic model that you illustrated with your little rope playing pieces, could account for some of that variability. And have you tried to look at centromere structures at different stages of the cell cycle? Whether
oh no, this is all from a cell line. Well, I can't say. So it's from lymphoblastoid cell line from Craig Venter. There's other lymphoblastoid cell lines that show the same thing, but HeLa cells also show the same thing. So, because we looked at those, at ChIP data from that. So I don't, and the other thing is that there's also this haploid cell line that was used in the Eichler study where we did the PacBio sequencing, and we basically see the same. We basically see the long arrays of the SEN1 line. So what we're seeing, I can say is general for the human genome. I think we have enough examples. That's epigenetic. Okay, so I was making a distinction and answered a question about neocentromeres, and they occur from time to time in the human population in the germline. They occur frequently in cancer. And those are epigenetic centromeres. Absolutely, I'm not denying that. I'm just saying when you look at the evolved centromeres, our natural centromeres, the ones that we're most interested in, that's not what we see. Thank you very much.